Hello, everyone. It's both a pleasure and an honor to be here recording for you, women, from the second Women Bioinformatics and Data Science LA conference. My name is Ana Carolina Heidenreich from Physiology, Molecular Biology, and Neuroscience Institute from UBA CONICET.
My name is Macarena Algañaras from the Center of Experimental and Applied Endocrinology in the University of La Plata, CONICET.
Here we are going to summarize our research work devoted to discover global transcriptional effects of Ingap-pp treatment on rat pancreatic islets. As an introduction,
Type 2 diabetes is characterized by both a peripheral resistance to insulin action and a limited capacity of beta cells to secrete insulin according to Eastman. This is due to beta cell loss of function and mass. Ingap is lead neogenesis-associated protein and also a derived pentatic peptide, Ingap-pp, is able to induce an increase of beta cell mass and potentiate insulin secretion in response to glucose. In this work, we analyzed the effect of Ingap-pp on gene expression of isolated rat cells exposed to it and how this change could be related to its non-physiological effects. Pancreatic islets were isolated with collagenase from normal adult male wistar rats divided into two groups and further cultured for 4 days in the absent control group or the presence of Ingap-pp. We performed 3 biological replicates and total RNA was isolated and sequenced from all the samples.
Figure 2 shows how public tissues and our control and Ingap-pp treatment has been used to perform a de novo reference annotation. In the part B of the figure, we show the standard pipeline of HiSat string type using all those files to generate the estimate of gene abundance. Figure 3 shows how linear mixed effect model has been used to account the differentially expressed genes. As results, in Figure 4, we can see how PCA help us to visualize these bulk samples in its transcriptomic signature to see similarities between our samples and the other public tissues. Figure 5 illustrates how rat pancreatic islets are composed of different cell types. Figure 6 exhibits a heat map of all genes clustered with hierarchical clustering to account selective expression variation by tissue. In Figure 7, we can see how is the variability between samples. We selected some specific genes to account the difference between expression into our control replicates. And those genes has been selected because they are specific of some lineage, cellular lineages. And we postulate that those samples were originally composed by different cell proportions. Figure 8 exhibits how many genes has been modulated by Ingap-pp treatment. Those genes has been detected by linear mixed effect model. We could detect 1,719 genes. Panel A shows how many modulates up and how many were downregulated. Panel B shows how many genes has been non-annotated. We discovered that 79 of Ingap modulated genes has not been annotated in the reference annotation from NCBI delivered on April 2020.
With 1,676 Ingap-pp upregulated genes, we perform a gene ontology analysis using Panther. The bar plot in the figure represents a significantly enriched pathway. Also, some of them have been previously reported to be regulated by Ingap-pp. We found a few novel ones, like RAS and WNT. Circus plots show which out of the upregulated genes belong to the mentioned enriched pathway, and you can see that some genes are involved in more than one pathway. In order to get the genomic profile that could help us to characterize possible function of these unannounced genes, we evaluate public data from chipset performed on a rat beta cell line in 1. We found some histone modification associated to active promoters, active enhancer, regulatory regions, and genes that are being actively transcribed. We further evaluate the coding potential and look for difference between eRNA, low non-coding RNA, or genes. This study is still ongoing, and we are currently evaluating the possible homology of this specific transcript with mouse and humans. In conclusion, we could say that Ingap-pp modulates gene expression of several mediators of signal pathway related to beta cell activation, myogenesis, apoptosis, insulin secretion, among others that could modulate beta cell function and mass. Besides, the finding of an unnoted transcript modulated by Ingap-pp represents a new challenge that we will further investigate to identify new targets that could be mediating Ingap physiological effects on rat islets.